In this lecture, I will talk about class 1 MHC molecule and how antigen are processed and presented by class 1 MHC molecule. But before beginning, I want to mention some basic facts about the class 1 MHC molecule. The first point is class 1 MHC molecule actually expressed by all nucleated cells. All nucleated cells. So those cells have nucleus can express MHC1 molecule. As we know that RBC, red blood cells, do not have nucleus, so this cell cannot express MHC1 molecule. And the MHC1 molecule is recognized by a subset of T cells called cytotoxic T cells. It is also known as CD8 positive cells because the cytotoxic T cells express a co-receptor that called CD8 positive. So this cytotoxic T cells have T cell receptor that can recognize the MHC1 molecule. And as I have mentioned, all nucleated cells contain MHC1 molecule, but the expression can vary. Like the muscle cell, liver cell express very low amount of MHC molecule or very undetectable level. But the cells like lymphocyte, B cell, T cell express huge number of class 1 MHC molecule. It can express one B cell or one T cell can express 50,000 of class 1 MHC molecules. So the expression of the MHC1 can vary, but all nucleated cells can express MHC1 molecule. The second important point is MHC1 molecule is composed of two polypeptides, that is alpha chain and beta 2 microglobulin. In one experiment, scientists have found that a cell called Dowdy cell, this cell is actually tumor cell. This cell can produce alpha chain of the MHC1 molecule but cannot produce beta 2 microglobulin. As the cell cannot produce the beta 2 microglobulin, the cell cannot express the alpha chain only. So to express alpha chain, the beta 2 microglobulin association is important. When scientists transfect the Dowdy tumor cells with beta 2 microglobulin gene, then the Dowdy cell can express the alpha chain and beta 2 microglobulin, means MHC1 molecule. From that experiment, they have found that both the alpha chain and beta 2 microglobulin is important for the expression of MHC1 to the plasma membrane. The third important point is, let me draw a MHC1 molecule. Let's assume this is a nucleated cells. This is the nucleus and this is the nucleated cells. This is the plasma membrane and the alpha chain looks like this, and this is the beta 2 microglobulin. So this is the alpha chain. This alpha chain consists of three domains. One is called alpha 1, alpha 2 and alpha 3. And this is the beta 2 microglobulin. I am writing only beta 2. So this alpha 1 and alpha 2 make a cleft or groove where the antigen bind. So here antigen bind, and let me draw a T cell, cytotoxic T cells. Let's assume this is a cytotoxic T cells, TC cells. It express T cell receptor that can recognize this area, and this T cells, cytotoxic T cells, as I have already mentioned, express a co-receptor called CD8. This CD8 molecule can recognize the alpha 3 domain of the alpha chain. So this is the CD8 and this is T cell receptor. Okay. So in that case, I can write that the alpha 1 domain and the alpha 2 domain actually make peptide binding cleft or groove where the peptide can bind. And also I want to mention another important fact about the alpha chain. This alpha 1 and the alpha 2 region of the alpha chain show polymorphism. In my previous class, I have already mentioned that the MHC molecules show polymorphism, but all the MHC molecule, all the domain of the MHC molecule do not show polymorphism. Only the peptide binding region of the MHC molecule show polymorphism. So the alpha 1 and alpha 2 region of the MHC molecule, class 1 MHC molecule, show polymorphism. The next important point is the size of the peptide. In case of MHC1 molecule, the peptide size is very limited because the space provided by this alpha 1 alpha 2 is small, so it can only accommodate 8 to 10 amino acid peptide. And especially I want to mention the most of the peptide that bind MHC I molecules is approximately or actually 9 amino acid. The range is from 8 to 10, but most peptide that bind with the MHC I molecules is 9 amino acid. Okay, and another important fact is the peptide that's called anchor residue or amino acid. So this peptide contain anchor residue or anchor amino acid that bind with this cleft region very specifically. So this anchor residue or amino acid present in the peptide N and C terminus. N and C terminus, both the area, N terminal, C terminal. So some amino acids are very specific in the N and C terminus of peptide that bind with the class 1 MHC molecule. I want to mention that all peptide that bind with the class 1 MHC molecule, the C terminus amino acids are mostly hydrophobic amino acids. So, but some basic amino acid can also be present. Basic amino acid can also be present, but the C terminus is mostly contain hydrophobic amino acid. So this is about the anchor residue. Anchor means it can anchor with the MHC peptide complex. The next point is, let's assume this cell is healthy. A healthy cell, a nucleated healthy cell, can express MHC I molecule. It is a MHC I molecule. When a healthy cell expresses MHC I molecule in the plasma membrane, it actually present antigen that is self-peptide. What happens? Our protein that present in the cell have a half-life. Most of the proteins half-life is 10 minutes. So after 10 minutes they get degraded. These healthy cells actually express that portion of the peptide by MHC I molecule. So this is called self-peptide. So when a healthy cell is expressing class 1 MHC molecule with self-peptides, and if TC cells, cytotoxic T cells, come close to that healthy cells, these TC cells recognize the MHC I and the self-peptides, and it recognize that this cell is healthy. So it will not harm the healthy cell. It can recognize the MHC and the self-peptide. It cannot harm the cell because it senses that this cell is healthy. But when the cell gets infected with virus, specifically I want to mention here cytomegalovirus, hepatitis B virus, and a virus like adenovirus 12. When these three viruses infect a cell, or if a cell becomes tumor or transform into tumor cell, the cell starts to produce low amount of or undetectable level of class 1 MHC molecule. They starts to stop the production of MHC I molecule. So these three viruses have a mechanism, escape mechanism of immune system. These three viruses actually can inhibit a protein synthesis called TAP. This protein is important for the antigen processing and presentation of MHC I molecule. So a virus infected cells do not produce or produce very little amount of class 1 MHC molecule. Now cytotoxic T cells recognize T cell receptor with the MHC molecule, but if there is no MHC molecule, these TC cells cannot sense that there is any infection, because to identify the antigen, the T cells require the MHC molecule. But if the infected cells produce low amount of MHC molecule, then the cytotoxic T cells cannot recognize the virus. So this is one mechanism of these three viruses to escape the immune system, so that TC cells cannot kill the infected virus cell. But there is another cell called NK cell. NK cell can sense when there is lack of MHC molecule. The NK cell can sense that this cell is infected, and NK cell kill the infected cells by the same way that is performed by cytotoxic T cells. So NK cell can recognize that these cells have some problems, so that it is not producing the MHC I molecule, because NK cell also secrete perforin and granzyme. Another last point I want to mention here, basic fact about the class 1 MHC, that is point 8. Some non-classical class 1 MHC molecules also present. Non-classical means it do not take part immune system actually very actively. The example of one non-classical MHC I molecule is HLA-G. In case of human, the MHC molecule is called class 1 MHC molecule, or MHC molecule is called HLA. HLA means human leukocyte antigen. So in mice it is called MHC, but in case of human it is called HLA. So one non-classical MHC1 molecule is HLA-G. What is the function of this non-classical MHC molecule? It has a very important function. So we are surviving. When a mother actually conceives a baby or fetus, the fetus have both the MHC molecule, it may be MHC 1 or 2, I am talking about MHC 1 because today's topic is MHC 1. So the fetus can express MHC 1 from the father chromosome and the mother chromosome. Okay. Now the fetus is present in the uterus of the mother. So it is the uterus. So when the fetus starts to produce mother MHC molecule, as I have mentioned that MHC molecules can be inherited co-dominantly. It means a fetus cell can express both the MHC molecule from father chromosome and mother chromosome. So now let's assume this is the mother MHC molecule, class 1 MHC molecule, and this is the father class 1 MHC molecule. These cells, fetus cells, is expressing both the MHC1 molecule. Now if these fetus cells express, sometimes the fetus cells can express paternal, means father protein, father antigen. So let's assume this is a father antigen. So a fetus is contacted with the uterus, its mother immune system. So the mother have cytotoxic T cells. The mother have cytotoxic T cells, and the fetus is expressing both the mother MHC and the father MHC with the father antigen. It can be mother antigen. If the MHC is presenting the mother antigen, there will be no harm. But if the fetus is expressing its paternal antigen like father antigen, then some problem can occur, because cytotoxic T cells can recognize the mother MHC, because these TC cells actually coming from the mother immune system. So this TC cell can recognize the MHC of the mother chromosome, but it can recognize that this protein is foreign. So the immune system of the mother can recognize this fetus as a foreign, because the protein is coming from the father, but the mother chromosome is expressing or presenting the antigen. So the TC cells can recognize the self MHC, so it can recognize the class 1 MHC molecule of the mother and the antigen of the paternal as a foreign. So the immune system of the mother recognize fetus as a foreign. Then what happens? The cytotoxic T cell will kill the fetus cells and no fetus can develop, but this is not occurring. So some molecule is actually responsible for inhibiting the cytotoxic T cells from killing. The example is HLA-G. This non-classical class 1 MHC molecule actually present in the interface between the fetus and mother. So if this is the fetus interface and if this is the mother interface, the fetus cells starts to express a non-classical MHC molecule that cannot be recognized by the cytotoxic T cells. So this is one example of non-classical MHC molecule that perform a very important role during the development of fetus. When this molecule represent paternal antigen, this cytotoxic T cells cannot recognize this type of MHC molecule. So fetus is not recognized as a foreign. So this is all about the basic concept of MHC1 molecules. Now I will talk about the antigen processing and presentation by the MHC1 molecule.